AS Biology. Welcome. Are you excited for a fun learning session? Make sure you have your book and a pen before we start. Today you are going to learn about cell fractionation. Cell fractionation. Fractionation is the process of splitting something up into fractions. Those of you who did chemistry at GCSE may remember the process of fractional distillation. Well, this is similar, in that cell fractionation splits the cell components into fractions based on their weights. There are three stages to cell fractionation. Stage 1 is called homogenization, and this means to break down the cells. Breaking down the cells means breaking down the cell wall and/or membrane to release the organelles. This can be done by grinding with a pestle and mortar, by mixing in a blender, or even by squashing by hand. During homogenization, the cells must be kept in a solution that is ice-cold isotonic. The solution must be ice-cold because this prevents the enzymes of the cells from breaking down the organelles too quickly. The solution must be isotonic. Isotonic means that the concentration of the solution is very similar to that of the cells. This prevents the organelles from being damaged due to osmosis. Stage 2. The solution is filtered through a gauze or fine mesh. This acts to remove the large parts of cell membrane and other parts of debris. Stage 3 is called ultracentrifugation. This is the stage that separates all the organelles from each other. This tube is placed into a centrifuge, and spun slowly. At low speed, the heaviest cell organelles fall to the bottom of the tube. The nuclei are the heaviest organelles, and so they fall to the bottom first, and here they form a layer known as a pellet. The liquid, known as the supernatant, is spun again and at a slightly higher speed, and so the next heaviest organelle will fall to the bottom of the tube and will form a layer on top of the nuclei. The supernatant is spun again and again at faster and faster speeds to separate all the organelles off into their separate layers. The organelles separate into layers based on their mass. The heaviest organelles drop down first, with the lightest dropping out of solution last. The order that organelles normally drop out of the supernatant and form a pellet is from heaviest to lightest. Nucleus, mitochondria, lysosomes, endoplasmic reticulum, ribosomes,